situation that will hamper our crop production worldwide. So mung bean, it's a kind of drought tolerant crop. That's why we have selected. So here you can see this is a mung bean, Vigna radiata, and it's a root nodule, leguminaceae family. And this is a from our experiment. It is a rhizobial, pure rhizobial bacterial colony, right? So these are the things I'm going to discuss today. And likewise, I have just told, so plant-based protein, demand of cheap source of protein is nowadays a greater concern. So in that category, plant-based protein, mung bean, it is widely cultivated and drought tolerant, and uh, summer crop mainly. And India, it is also cultivated in rainy season, that is Kharif, and it is a source of around 27% protein. Apart from protein, there is carbohydrate, iron, folic acid is there. So there is a major component of our diet. So uh, this mung bean, if we reduce the abiotic stresses, so we can increase the production. Even India also very much concerned about mung bean production. I will come. So mung bean as a plant, if you see botanical point of view, it has a strong root system architecture that also determines the rhizosphere microbial colonization also. So when we are focusing on the root nodule bacteria, so their source is from the bulk soil. Then they will enter in their life cycle and they fix nitrogen and produce nodule. So this mung bean crop and its associated nodule bacteria rhizobia, it can fix around 58 to 109 kg nitrogen per hectare per year. So that much of capacity is there. But if you see in India, so India's total contribution is higher, 54% worldwide. Average global productivity or yield is 730 kg per hectare. But India's productivity is very less, 500 kg per hectare. It is far reduced, right? Even due to arid region where rainfall is less than 250 millimeter per year. So there, like Rajasthan, Gujarat, so their mung bean production is around only 300 kg per hectare. So that's a bigger concern. So now you can compare. So 730 and this is 300 only. So day-to-day soil moisture deficiency is increasing. So many reasons are there. Global warming is there, increased temperature, and infrequent monsoon rainfall. And mostly mung bean production is depend on rainfed. It is a rainfed. So the irrigation purpose, the water mostly farmer depends on the rainwater. But if there is frequency of rainfall reduced day to day, so drought stress, that mung bean crop suffers havoc drought stress. So mung bean productivity, either vegetative or reproductive yield. So our concern is protein content or the number of pods, right? So it is drastically reduced. So these stress factor also have significant influence on the performance of beneficial microorganisms, either rhizosphere or phyllosphere or the soil microbial communities, right? So unlike legumes, rhizobia have high affinity to tolerate drought. So that's why we are selecting this nodule bacteria rhizobia, because rhizobia has the same kind of means drought tolerant adaptation. So it helps to plant to increase more drought tolerant. So application of plant growth promoting rhizobial strain or nodule bacterial strain, it's a major strategy to improve crop productivity under water deficiency. So this strategy is recent days it has very much impact. So apart from this strategy, we have other strategies also. If we develop drought tolerant crop, but we need a long-term period, because there is multiple genes are associated, complicate gene regulatory network are associated. If we are improving drought tolerance a particular crop, we are increasing or enhancing their drought tolerance. Suppose mung bean already drought tolerant, but nowadays situation are again, it is creating more drought stress in soil moisture stress, right? So in that condition, that mung bean also suffers. So if you are increasing the drought tolerance in variety developmental program, so it depends on long term. But for application of plant growth promoting rhizobial, it has many benefits. So it can be a good component for dry farming, and also it helps to make more drought tolerant and eco-friendly way, right? So that's why we are taking this objective to identify the bacterial strain from mung bean root nodule. It has also plant growth promoting traits. Different plant growth promoting traits are there, I will discuss. And it also survive moderate to severe drought condition. So we have tested in pothouse also. So apart from in vitro testing analysis, we have tested in pothouse also. And this is the pioneering study in mung bean, French bean, and other crops are reported. But for mung bean, this is the first study. So coming to methodology, as most of the things are known, it is taken from literature. So we have selected the higher affected arid region, particular arid soil. So haplocalcid arid soil type where carbonate-based salts are there, and mostly 4 districts are there. So Churu, Bikaner, Jaisalmer, and Barmer. So we have selected 34 sites. So the sites, so these are our collection sites. So it is a larger area, around 500 miles. So they are desert region, arid soil, and spanning around 500 miles, covering 4 districts, Churu, Bikaner, Jaisalmer, and Barmer. And we have collected nodule samples from different plots and also rhizosphere soil. So rhizosphere soil, we have analyzed many physicochemical parameters, pH, EC, organic carbon, total nitrogen, right? So am I audible? Any disturbance is there?
You are audible, no disturbances. Please go ahead.
Okay, thank you. So then we have isolated rhizobia from nodule samples. There is particular media is there, yeast extract, mannitol, agar media. And after isolation, we are maintaining that culture. And then we go for elite strain, how we can screen out for elite drought tolerant strain. So based on abiotic stress tolerance, first we have given drought tolerance by addition of polyethylene glycol 6000. So different concentration we have analyzed 10%, 20%, 30%, 40%. So we have taken 30% and 40%. And then we go for temperature. So why we have taken temperature? Because in natural condition, there is drought also there and simultaneously temperature also there. So that's why we are taking combined stress tolerance. So apart from separate stress tolerance, now we are also taking combined stress tolerance. So either 30% polyethylene glycol 6000 and temperature is 40 degrees centigrade, or higher drought tolerance 40% PEG 6000 and higher temperature 45 degree centigrade. So these are just analyzing their stress tolerance of nodule bacteria. Now for nodulation, we have studied the nodulation genes, NOD-C genes, by amplifying specific gene markers using PCR. Then we have studied the plant growth promoting traits in drought condition also, without drought condition also. So here different parameters we have studied. So free living nitrogen fixation ability we have studied. Here gas chromatography analysis we have performed. Phosphate solubilization, qualitative and quantitative both. So we have used special type of media by addition of tricalcium phosphate, NBRI media, National Botanical Research Institute, modified form we have used. Mineral rock phosphate we have added, the same thing, tricalcium. And also we have quantitatively estimated what is the phosphate actually solubilized a particular time period. So temporal variation also we have checked. So apart from this, indole acetic acid, auxin hormone production, ammonia production, and ACC deaminase activity. So drought tolerance, particularly rhizobia and nodule bacteria. So already background study reported ACC deaminase activity. It is a kind of mechanism that give the drought tolerance to particular rhizobia, and it helps to plant and making more drought tolerant. So that's why we have included this type of plant growth promoting traits. Now we have studied their symbiotic performance, this allied nodule bacteria. So here we have conducted water deficiency experiment in different field capacity. So we are actually analyzing their capacity, how much efficient in the terms of symbiotic performance, nodule number, and also biomass, their dry matter from the control. So we are actually doing the moisture content in different soil moisture regime, 100% field capacity, there is no drought, 50%. So we have reduced the irrigation or water content. So 50%, then also again, approximate 25% field capacity. So now it is severe drought. So there is moderate drought is 50%. So 50% water we are not providing, right? So 25% again, it is half. So it is severe drought tolerant condition. So this type of reducing soil moisture percentage we are creating artificially, because recent days this type of situation is occurring actually. So recent days concerned, we are making same type of environment in pothouse, and our allied nodule bacterial strain we are employing, and we are trying to improve the symbiotic performance, their nodule dry weight and also fresh weight, and their biomass, root weight, shoot, dry matter, so everything. And lastly, identification based on their ribotyping 16S rDNA gene sequencing. So what is the actual rhizobia or any other bacteria is present, we are just identifying, right? So apart from statistical analysis, it has a major role. So here we are employing such kind of statistics so we can extract more data that is not still reported. So here we have analyzed multivariate statistics. We have seen our data is not normally distributed. So apart from ANOVA or simple t-test, so we are employing here environmental effect. So there is multifactorial effect, different types of effect is there, different types of PGP traits are there, even soil factors are there. So Parmanova test we have performed to know the significant difference in vitro drought tolerance of particular allied bacterial strain. Either it is 20%, 30%, 40%. Isolated from different district or different arid regions. So how they are significantly varied, in how much varied? We have just extract these things. So this kind of analysis is first time. So apart from Parmanova, we have used network analysis also and ANOSIM analysis. So second Parmanova test also we have employed based on their combined stress tolerance. So first one is only drought tolerance, and second one is combined stress tolerance as a variable and different plant growth promoting traits. So how much they are different, whether it is originated from sampling district or it is significantly varied from different bacterial isolates, right? So likewise, just I am giving this data. So how much pH? So it is highly alkaline soil. So as I have told, it is suborder is haplocalcid. In erythritol, haplocalcid order is there. So carbonate based calcium carbonate based salt is there. So this district Churu, so pH is around 7.9 and maximum 9. Even Bikaner Jaisalmer also 9.1. So texture is mostly sandy soil. So if you see electrical conductivity, so maximum 0.45. Organic carbon is very less. So 0.12%. So compared to any organic soil or a normal neutral pH soil, so this organic carbon content is very less. Even total nitrogen also deficient. So if you see, this is the total data. So here coordinates number of plants. So 5 plants is excavated from each field. Average number of nodules studied. So total around 400 nodules are studied. And total nitrogen content available, phosphorus K. So why this is matter? These things also correlated with the survival of the rhizobia. And this is the collection site already I have shown. So now here 34 sites are there, arid sites. Total plants studied 170 and 400 nodules and total number of isolates 98. So this is also a picture. This is plates of yeast extract Manita lager, where you can see the transparent colonies and just dew-like appearance. So this is the rhizobial colonies, pure rhizobia. So when we go for drought and temperature tolerance study, So here you can see this is the number of isolates, and when you go for 40% peak tolerance, so Barmer there is no culture is showing 40%. So if you correlate these things, Barmer also you can see their pH also less, 7.7 to 8.3. But when pH is, it is highly alkaline. So drought tolerance also increased. So such kind of study we have performed. Now also this study is going on in different types of crops apart from mung bean, other legumes also. So you can see this data. So here you see there is constant reduction of the bacterial